plant hormone. Communication in plants. Communication between cells in different parts of plants is required to coordinate the direction and time of growth, water balance, other plant responses. Plants do not have any nervous system, so internal coordination is controlled by hormones. Germination. Germination is how a plant grows from a seed. It is a process which a dormant mature embryo sporophyte in a seed resumes growth. Certain species-specific conditions may be required to break dormancy. It begins when water activates enzymes in the seed. It ends when the embryo breaks the seed coat. Growth. Growth is an increase in cell number and size. It occurs primarily at meristems. Differentiation results in the formation of tissues and parts in predictable patterns. Patterns of plant development are an outcome of gene expression and environmental influences. Plant hormones and other signaling molecules. Plant development depends on cell-to-cell communication mediated by plant hormones. Plant hormones, they are the signaling molecules that can stimulate or inhibit plant development, including growth. There are 5 types: Gibberellins, Auxins, Abscisic Acid, Cytokinins, and Ethylene. Hormonal responses. Responses in plants are simple, no equivalent to endocrine system of animals. Hormone-producing cells in plants are not organized into specialized tissues such as glands. They are generally produced by the cells receiving the appropriate environmental stimulus. Responses are slower than in animals. Hormones are distributed through the plant in a variety of ways: cell to cell, through transport pathways, usually phloem, and through the air. Gibberellins. Gibberellins induce cell division and elongation in stem tissue and are involved in germination. Can speed germination in spring by overcoming seed dormancy and the requirement of light. It can cause the formation of giant flowers. Treating seedless grapes with gibberellin produces larger, juicier fruit. It can be used to prevent root growth in potatoes, thereby preserving the crop. Gibberellin was first recognized in 1926 by Japanese scientist Eiichi Kurosawa studying Bakanae, the foolish shielding disease in rice. This disease is caused by the fungus Gibberella fujikuroi, the metabolism of which produces a surplus of gibberellic acid. Growth is to be taller, thinner, and having no rise in the husk. They are incapable of supporting their own weight, they topple over and die, hence they are called the foolish shielding disease. Auxins. Auxins promote or inhibit cell division and elongation depending on target tissue. Auxin produced in the shoot tip prevents the growth of lateral buds, which is also called apical dominance. Auxins also induce fruit development in ovaries and lateral root formation in roots. Spraying auxins can 1. Prevent natural pollination, 2. Produce seedless vegetables such as tomatoes and cucumbers, 3. Prevents fruit fall by delaying abscission, and 4. Induce flowering in the pineapple family. Auxin causes depending on plants. Auxin is evenly distributed throughout the tip and the coleoptile grows straight out. If light is concentrated in one side of coleoptile, then the auxin moves away from the light source to the darker side of the tip and becomes more concentrated in the cells of that region. The increased concentration of auxin in these cells means they grow more quickly than cells that is nearer to the light. The uneven growth of cells results in the bending of the coleoptile. Auxins, the bad. Auxins are both stimulators and inhibitors of growth. Synthetic auxin-like chemicals 2,4-D and 2,4-T were used as herbicides. Both contain trace levels of the auxin as contaminant. The combination of these two chemicals were referred to as Agent Orange during the Vietnam War and was used as defoliant as it causes such rapid, disproportionate growth that leaves or treated plants shriveled and died. At the correct concentrations, these chemicals are selective for broadleaf weeds and do not kill grasses. Abscisic acid, ABA. Abscisic acid or ABA inhibits growth. It is part of a stress response that causes stomata to close and diverts product of photosynthesis from leaves to seeds. It is produced in terminal buds during winter, slows plant growth, directs leaf primordia to develop scales. It antagonizes gibberellin in seed germination. Cytokinins form in roots and travel to shoots, where they induce cell division in apical meristems. Cytokinins also release lateral buds from apical dominance and inhibit leaf aging. Volkskuhl discovered the effects using coconut milk in the 1940s at the University of Wisconsin Madison. Adding coconut milk to plant tissue culture causes it to regrow vigorously as a callus. Hence, you can regenerate an entire plant from just a little bit of tissue due to cytokinin content in coconut milk. Cytokinin acts in concert with auxin. The two are complementary, having generally opposite effects. Ethylene. Ethylene is the only gaseous hormone. It is produced by damaged or aging cells. It induces fruits and leaves to mature and drop. It is also used to artificially ripen fruit. The change of a fruit before and after ethylene. So the fruit starts as acidic, but with ethylene, kinase is activated and it turns it into neutral. Aminase is also activated by ethylene, turning starch into sugar. Hydrolase changes chlorophyll to anthocyanin, which makes a fruit from green to red. Pectin, it is broken down by pectinase, which is also activated by ethylene, into less pectin, which makes the fruit soft. And large organics within the fruit will be broken down with hydrolases, which causes them to become aromatic carbon instead. Ancient Egyptians gas figs in order to simulate ripening. So ripening, it is caused by wounding. So the wounding simulates ethylene production by plant tissues. The ancient Chinese burned incense in closed rooms to enhance the ripening of pears. Besides hormones, other cellular molecules are developed in plant development. Besides hormones, other cellular molecules are involved in plant development. For example, brassinosteroids, FT protein, salicylic acid, histamine, and jasmonids. Examples of plant hormone effects: Gibberellins and barley seed germination. Barley seeds absorb water. Embryo releases gibberellin. Gibberellin induces translation of amylase gene. Amylase breaks starch into sugars used by embryo for aerobic respiration. Auxin or IAA plays a critical role in all aspects of plant development. So auxin, it is involved in fertilization of zygote, polarity and tissue pattern in embryo, formation of plant parts, differentiation of vascular tissues, formation of lateral roots, and responses to environmental stimuli. Jasmonid signal plant defenses. Wounding by herbivores will cleave peptides such as histamine in mesophyll cell. As you can see, the caterpillar is actually eating the leaf which wounded it, and it will release jasmonid. Activate peptide stimulate jasmonid synthesis, which turns on transcription of several genes. Some gene products slow growth. Other gene products signal wasps to attack a specific herbivores responsible for damage. Adjusting the direction and rates of growth. Tropisms. Plants adjust the direction of rate of growth in response to environmental stimuli such as gravity, light contact, and mechanical stress. Hormones are typically part of this effect. Gravitropism. Gravitropism is a growth response to gravity which causes roots to grow downward and shoots to grow upward. Cytoliths, they are amyloplasts containing heavy starch grains that sink to the bottom of the cell. A change of condition results in movement of cell auxin efflux carriers. Phototropism, orientation of certain plant parts toward the light. Non-photosynthetic pigments such as phototropins respond to blue light, initiating signal cascades. Auxin is redistributed to the shady part of the plant. Thigmotropism, contact with a solid object changes the direction of plant growth. Involves touch genes and calcium ions. Results in unequal growth rates on opposite sides of the shoot. Mechanical stress such as wind inhibits stem lengthening in a similar touch response. Sensing recurring environmental changes. Seasonal shifts in night length, temperature, and light. Seasonal shifts will be triggered in plant development. Flowering plants respond to recurring cues from the environment with recurring cycles of development. Biological clocks. They are internal mechanisms that govern the timing of rhythmic cycles of activity. You have circadian rhythm and solar tracking. Circadian rhythm is cycle activity that recurs every 24 hours. Solar tracking, it is a circadian rhythm in which a leaf or flower changes position to continuously face the sun. Setting the clock. Different wavelengths of sunlight set biological clocks by activating photoreceptor pigments called phytochromes. Active phytochrome causes gene transcription for components of rubisco, photosystem 2, phototropin, and molecules involved in flowering, gravitropism, and germination. Photoperiodism. Long day plants flower when nights are short. Short day plants flower when nights are long. Short day, for example, tobacco, chrysanthemum, strawberry, soybean, and rice. Long day, such as spinach, lettuce, clover, and greens. There are some plants which are day neutral, not affected by daylight. For example, tomatoes and cucumber. Leaf cells transcribe more or less of flowering gene in response to changes in length of night relative to the length of day. Long day plants would have high levels of PFR that may simulate foragen secretion. Short day plants, foragen may be secreted when PFR levels are low. So this is an experiment showing that it is the length of darkness, not light, that controls flowering. In A, you can see that the light is longer than dark. Light is 18 hours, dark is 24 hours. 6 hours. 24 minus 18. So for short day plants, no flowering. Long day plants, they flower. Okay. In B, we interrupt the middle of the day with a little bit of darkness. So at hour number 6, we suddenly turn it dark, and then we turn it light again. So for short day plants, it is still no flowering. But for long day plants, despite the interruption, they flower. In C, we have 6 hours of light, 18 hours of darkness. So short day plants will flower, long day plants will not flower because it needs a long day. And in D, as you can see, the day is short, the night is long, but in the middle of the night, we interrupt it with a brief period of light. So first of all, you have 6 hours of darkness, and then a little bit like 8 hours of darkness, and then a little bit of light, and then back to darkness. Even with this small amount of darkness, short day plants cannot flower because the darkness is not enough. But for long day plants, they're able to flower because there is less darkness and much more light for it to flower. Flowering and vernalization. Vernalization means the flowering period per year. So some biennials and perennials flower in the spring, only after exposure to cold winter temperatures. Senescence and Dormancy. Senescence is the phase of a plant life cycle between full maturity and death of the plant of plant parts. Abscission. It is a process by which plant parts are shed. It is triggered by many factors, including seasonal changes in environmental conditions. In deciduous plants, abscission usually happens during midsummer and autumn. Midsummer, Auxins is produced. Plant diverts nutrients to flowers, fruits, and seeds. So during midsummer, some of the leaves will fall. In autumn, autumn production declines in leaves and fruits. Ethylene signals enzymes to digest cell wall abscission zones, which causes leaves and fruit to drop. Dormancy. A period of arrested growth that is triggered and ended by environmental cues. Signals to begin dormancy include long nights, cold temperatures, and dry nitrogen poor soil. Favorable conditions signal to break dormancy. As you can see, this is a Venus flytrap plant. Both of these are the same plants, taken at different times of the year. During dormancy, they are much more smaller, kind of less active, but during the growing season, they are much more lively looking, and then they are much bigger. The end.